Assalamu alaikum wa rahmatullahi wa barakatuh. Dear all students, I wanted to discuss with you today a very interesting topic, which is the mycology or mycosis, which is mycology infections. And as we learned in this course, only a single lecture of mycosis, but you have to bear in mind that mycosis or diseases due to fungi are very important and very significant. And the fact that we only have one lecture in this course does not mean that they are not very common. Actually, they are very common. And you might be seeing a lot of fungal infections throughout your practice or throughout your um, work in the laboratories. So as you learned from the lecture, mycosis is the study of fungal infections, and we call this branch as mycology. And these fungi are ubiquitous in nature. They are found in humans, in animals, in soil, and in the soil they are important because they degrade any organic compounds, and some of them can produce also antibiotics. Like we learned, the, the penicillin antibiotics are actually synthesized by what do you call it, fungi. And they are very important because these compounds inhibit the growth of competing bacteria. And there are only few of hundreds of thousands of species of fungi that are pathogenic to humans. And I've learned throughout throughout the whole years of my practice that every year we see a larger number of yeast that are becoming more pathogenic, and also a new type of fungal infections emerge every year. And over the last 3 decades, or maybe 4 decades, we are seeing unprecedented increase in the diversity of life-threatening invasive fungal diseases. And this is parallel to corresponding increase in immunocompromised patients, because there's increase in HIV infected individuals or some individuals undergoing immunosuppressive treatments. So that was a summary of the previous lecture. So let's proceed with the cases. And it's, as I said, it's very common to see fungal infections, even though it's one lecture. So please bear in mind that what is presented in this practical is only a few cases of what we actually see on practice. The first case is a 5-year-old boy, post-bone marrow transplantation, developed fever. He is non-responsive to antibiotics, and the blood culture grew this yeast. So what do you call this organism? Let's first of all assume that once you receive a blood culture, you immediately think of bacteria, because whatever grows in a blood culture is usually bacteria. However, Candida is the exception, which is a yeast, not a bacteria. And actually we can grow it from a blood culture, and also we can grow it on a blood agar. So this is yeast, and we can easily distinguish it on the blood agar from the appearance of the colonies and the fact that these colonies have very distinctive characteristics. So by the time that bacteria starts growing on blood agar, you can see that they grow within, let's say, less than 24 hours. But for yeast, usually it takes longer, and the colonies appear differently on a blood agar and also under the microscope, like we will see in a minute. So you can also, beside blood agar, you can also, if you're suspecting yeast growth or a fungal growth, you can grow it on a standard media for yeast, which is called sabrodextrose agar, or SDA. So this is very specific media for yeast because it has all nutritious substances that can stimulate the growth of this type of infections or yeast. And the typical characteristics of these colonies are different from those of bacteria, because you can see creamy whitish colonies that have either flat or dome shape, and usually can see them very waxy, or they have either dry or waxy surface, but usually we see waxy surfaces on the top of the agar. And once we see that, we immediately know that this is yeast. But again, I remind you that yeast usually takes up to 72 hours for growing at 37 degrees. And this is the same as internal body temperature of a human. So what we usually do in the lab, if we want to encourage the growth of this yeast from the plate, we can keep it at room temperature for another 24 hours, and what you see the next day is very clear, beautiful colonies on the agar. So after suspecting yeast, we need to confirm. And the idea of doing additional tests, although we are a little bit, or we can say that almost 100% sure that this is yeast, because we want to take it to the next level, which is identification at the species level. And this is an additional test that we do, which we call germ tube test. And the principle of a germ tube test is to incubate the yeast in the human serum. So we have some serum in the lab, and we incubate the candida or the yeast in that serum. So how do we incubate? You incubate it for 3 hours at the room temperature, at 37 degrees, which is an incubator temperature, and then we observe the germ tube. This germ tube signifies the growth of hyphae, and they emerge from the yeast-like structure. I'll show you in a minute. So let me use the pen. You will be able to see that this is the mother cell here, and there is some germination coming out. So there is narrowing of the growth, and there is a long tube. We call it germ tube. You can see also here budding yeast, which is a daughter cell that is coming out of the original mother cell. And this is very typical of the species Candida albicans. So what is the importance of this test? The importance of this test is that it differentiates between the different types of Candida species, because there are different species, a long list of species that I don't want you to memorize, but you need to remember that Candida albicans produce the typical germ tube, which is about half size and half width of the original length of the mother cell, which is 3 to 4 times the length of the mother cell. And there's a restriction, like I showed you here. There's a restriction at the neck. And this is the initial stage of hyphae, because initially it will form this germ tube, and then it will keep branching and branching to produce the hyphae that we learned in the lecture. This will differentiate between the hyphae and the pseudohyphae, which is produced by other types of species called Candida tropicalis. And how do we know that this is pseudohyphae, not a real hyphae? By the restriction at the neck. So if there is no restriction, then this is another species of Candida, which is tropicalis. However, we have to bear in mind that sometimes there are some species of Candida that produce this germ tube with its restriction, and we call it Candida duplinensis. So we have a long list of species actually. So at this level, we need to do additional tests to confirm that this is Candida albicans, and luckily in the laboratory we have automated system, which we call it Phoenix, and also we have MALDI-TOF, which will tell you that this is Candida albicans at a very species level. And I wanted also from you to remember that these cells are very commonly seen from a blood culture from individuals who are immunocompromised, like in this patient who has gone through transplantation. So he had been on immunosuppressive drugs. So that's why Candida, which is a usual or a commensal on the skin, can now invade the blood and also cause active infection in the blood, we call it candidemia. Okay, now let's proceed to the second case. This is a 35-year-old male attending the emergency department with 2 weeks history of on and off fever. So this basically means that he has been having what we call intermittent fever, which is not continuous, and he had severe headache. On examination, he had IV drug marks on his arms, and he had oral thrush. So oral thrush, I showed you the slide in the lecture which shows a growth of a white coat on the tongue and the mucosal surface as well. And then blood and CSF samples were collected, and the blood sample confirmed HIV positive results. So actually we don't confirm HIV result from a standard blood test, because this is highly suspicious to have HIV. That's why he has been tested for it, because of the IV drug marks. So you already know that you have a risk factor that this patient might have HIV. Then there is a co-infection, because HIV is a disease that can invade the immune system and cause the person to be immunocompromised and susceptible to different types of infections. So CSF was collected because this patient had severe headache, so cerebrospinal fluid was collected, and it grew something. So if you have a young patient with IV drug abuse and he has been confirmed to have HIV, you are highly suspicious that this patient is having an infection called Cryptococcus neoformans. So that's why we do a very specific stain or special stain, we call it India ink, to stain the capsule of this yeast. And I'll show you the slide in a minute. So this is what you see on an India ink. Very clearly you can see that there is some yeast here. Let me show you. So you can see this is the original cell, and this is budding cell coming out of it. And each one of these cells is surrounded by a halo around it. So this is what is called destaining, because the ink will stain the background and it will leave a halo, a capsule that cannot be stained by the India ink. And this is very typical of Cryptococcus neoformans. We always keep this infection in mind when we are suspecting meningitis or meningitis in immunocompromised individuals, not in a normal person, or not in a person who has an intact immune system. So please bear in mind that Cryptococcus neoformans is a clinical disease that we find in immunocompromised patients, and this condition is called cryptococcal meningitis, and it's very common in AIDS-defining illness. So what are the characteristics? So it's a yeast cell with irregular size, and there is a thick polysaccharide capsule. But you have to bear in mind some of these cells are not encapsulated, but the majority are, with one or more budding or buds attached to it. And how do we stain it? Actually, we see this on a clinical material, but not in the culture. So what do you mean by this? It means that if you take a CSF sample from the patient and you stain it, you will be able to see immediately the stain. India ink, you can see the capsule from the stain. So this means that from the original clinical material. But if you grow the Cryptococcus on a plate or a culture, you are less likely to see the capsule. There's a very rapid test that we use in the laboratory to identify it from the CSF. We call it latex agglutination test, which means that you use red blood cells that are coated with latex, and then it's very specific bound to Cryptococcus neoformans, and it forms an agglutination reaction. So it's the same principle to any lattice agglutination, but we use very specific antibodies. You have to also bear in mind that Cryptococcus neoformans does not only cause meningitis, it can cause a lung disease, we call it lung granulomas, that also occur in immunocompromised patients, and sometimes it resolves spontaneously in the majority of the patients without dissemination to other organs. And if it spreads, it has to spread through the blood. That can result in disease later on. What is the source of Cryptococcus neoformans? As you saw in this patient, he's an HIV drug abuse, so the contaminated needles must have caused this infection. So it comes from the environment or the surrounding environment and the contamination in the needles. So this is another picture showing you the Cryptococcus neoformans with the typical CSF appearance. This is another patient who is a 28-year-old HIV positive patient, and you can see here the teardrop appearance, which is very typical of Cryptococcus neoformans, and this happens at the very beginning of the formation of the bud. This is in B, you see a different type of stain, which is going to again highlight the capsule here, and it's a red stain or a mucin stain. How do we treat this condition? Of course, by now you have learned the antifungals. So we use intravenous amphotericin B. Sometimes we combine it with another antifungal, like for example fluconazole and so on. So with this type of severe infection in the brain, we need immediate treatment. So what is the appearance of a Cryptococcus meningitis in HIV patients? You can see here that there is very wet and mucoid colonies that you can see on a plate. Sometimes if you also prolong the incubation, you can see it turn into a darker color, and also it can sometimes also form colonies that are similar to candida, that are very creamy colonies on the sabrodextrose agar. Okay, now let's move on to the third case. So this is a 50-year-old lady with chronic otitis externa, which means it's a chronic disease that has been going on for a while. Otitis externa means an infection in the outer ear. An ear swab was collected and it grew the following organism. We want to describe it microscopically. So please bear in mind, any fungal infection we need to identify, we always rely on the macroscopic and microscopic appearance of it for identification and for the purpose of the treatment. In mycology, macroscopic means the appearance on the culture plate, and the microscopic means the appearance of the fungi under the microscope. So how do we describe this? So what you see here, this is the sabrodextrose agar. You can see here this black center, and you can see the white rim surrounding it. And you can see here also on reverse. So from the bottom you can see the reverse, where you see very tiny dark circle, or sometimes it's puff or yellow gray surrounding the white rim. We call this fungi Aspergillus niger because it produces very dark pigment or plaque pigment from the conidia. Usually the conidia matures from the center of the colony, and it grows, takes some time to grow. So it takes up to 72 hours to grow at 30 degrees Celsius. And you can see the surface of a mature colony is usually covered with a dense aggregate of a jet black conidium. And the reverse is what you see on the right side. I wish you could see this on live because we usually demonstrate this and it gives very nice appearance on the plate, something that you don't see in the bacteria. So under the microscope, after identification, macroscopically, we need to see it under the microscope. So what we do here is that we take just a little bit of that big growing colony on the plate and then we put it under the microscope after we treat it with certain agents. And this is what you see. You see black conidia extending from the vesicles. So this is the vesicle here, and these are the conidia. So what you see here is conidia, these small dots here, the black dots here. Okay, so now let's move on to the fourth case. So this is a 12-year-old boy, schoolboy, who has pets at home complaining of hair fall and itching in his scalp. So we took a scraping from his hair and then we want to see the macroscopic appearance. So this is very common. We call this type of infection dermatophytes because it occurs on the hair. And it's very common in children or people who have pets at home. So the source in this case comes also from the pet or the animal. This is what you see macroscopically. So first you grow it on the saprodextrose agar, and this is the appearance of the colony. What you see here is a white fluffy colonies on with radial grooves. So what we mean by radial grooves are these small lines, whitish lines extending from the center. Reverse is the bottom. What you see here is yellow, deep yellow color from the bottom of the plate. So this is Microsporum canis, the name of this fungi, and it's very typical, as I mentioned, from dermatophyte coming from animals. So again, we take a small amount of that colony, we treat it under the microscope to observe these microscopic appearance, and this is what you see. Why do you see this the blue stain? Why do you see it in blue color? Because we stain it with a blue stain called lactophenol stain. And the typical appearance is macroconidia. What we mean by macro means big conidia, typically forming what we call spindle-shaped cells. So this is the spindle shape. So this is what you see here is a spindle shape with these cross septations. Can you see them? And it has very typical tapered ends, which means that in the center it's very thick and wide, and it's narrow at the center. This is also having thick walls with terminal nopes. This is very typical of Microsporum canis. Usually you see very numerous, means large in number. They are very rough and they have very thick walls. Sometimes you can also see microconidium here. So it's not all of them having micro, they have a macro. There are also some microconidium. And what microconidium means, they are hyphae that are small, regular with some branching. And this is rectangular arthrospores, which means that they are divided into small, like here. See, this is a typical arthrospore. It's similar to the knuckles of your fingers, so it's divided into like 2 or 3 parts. Sometimes they can be also seen found in the skin, nails, or hair scrapings. So this is common in the keratinized layers of the skin. So you have to bear in mind that there is a differential diagnosis, which means that if you are suspecting this type of fungal growth or mold, you have to bear in mind that there are also other similar fungi causing similar type of infection. So we divide them into three: dermatophyte group, and there's the trichophyton species, and also the epidermophyton species. So there are three different types similar, and I'm not going to go through the details because there are so many details in these. It needs separate lectures that we do not have in this course. But please bear in mind that mycology is a very large branch of microbiology, but this is just simple. We're touching on simple topics very superficially. So let's say that you are now suspecting a fungal growth. You took a scraping. So let's say that this boy had some infection on the scalp. So you took scrapings from the scalp or from the hair. What we do in the lab is that we treat it with KOH, which is potassium hydroxide. We prepare it because this solution will help to remove any debris or dead skin and without compromising the fungal growth. So we place it under the microscope, and we should be able to see two types of growth, either ectotrix or endotrix type of invasions, which means that if the growth under the microscope shows invasion in the outer shaft of the hair, like here, this is the shaft of the hair center, and you can see it on the outer part, then this is ectotrix. If you see under the microscope growth of the fungal infection inside, like here you can see inside the hair shaft, we call it endotrix. And this tells us different types of fungi. You don't need to learn the details of like Microsporium gypsum and so on, but you need to remember that these are the different appearances. On the left hand side, this is another illustration which explains the ectotrix and the endotrix. So how do we diagnose this type of infection in the clinic? Let's say that you have a patient and you are suspecting this fungi or the mold growth on this hair, but you don't have the tools to it. The easiest thing to do is to send this patient to a dermatologist. They have what we call Wood's lamp, and this is a fluorescent lamp that you object it on the superficial layer of the hair, and you can see it shining like greenish to silvery fluorescent stain. And this is typical appearance of the ectotrix of Microsporium, and you can also see different types of fungal growth using this lamp. Of course, we don't have this lamp in our clinics like the GP clinics, but we have it in the dermatology clinics. It's a very easy, but it's a very useful tool also to diagnose different fungal infections. Okay, so this practical has came to the end, but I wanted you to just have a simple like quiz to take. If you stain fungi by gram stain, what do you think the gram stain would be? So I used to ask the students when they are in the lab, but since we are not doing this live, it's a gram-positive stain. So sometimes if you have a blood culture and you're taking a drop of that blood culture and examine it under the microscope with a gram stain, you can see gram-positive yeast. But you can easily say that this is not bacteria, this is yeast, because of the appearance and the size of the gram-stained cells under the microscope. This is what you see. They are usually larger than the bacteria. They have this budding appearance. Can you see the budding cells here coming out? I try to show you here. So these are cells with budding. This is very typical. As you can see, it's purple in color. It's gram positive. Another question. So, what is the scientific name of brewer's or baker's yeast? So, what is baker's yeast? It's the yeast that we use to bake bread or cake or biscuits at home. We add some kind of a yeast. So, this is just to remember that yeast are very useful in certain food microbiology applications. So, we use it to ferment the wheat and ferment the grains. So, you get this nice fluffy bread because of the action of the yeast, and we call this yeast Saccharomyces cerevisiae. You can also go back to the label on at home and check. This is the name of the fungi that we use to grow to ferment the bread. Now there is a very interesting information for you here also to remember. There's always a pleasant smell of rain, and you always wonder where this pleasant smell coming from, and actually it's because of a certain type of fungi that is very similar to bacteria. We call it actinomyces, and this is the actinomyces that gives the soil its earthy aroma. This is the appearance under the microscope. This is the end of this practical. Thank you very much for attending and listening to this practical. If you have any questions, please drop me an email or a message in the band group, and keep learning using your memes or whatever tools you have. Thank you very much.